Thanks, Jeremy. It's a great pleasure to talk at this meeting. So, as Jeremy said, Apitope is a spin-out from Bristol, and this is what we do. And towards the end of the talk, I'm going to be talking about one of our peptide cocktails that we're in fairly advanced clinical trials with at the moment. So the messages that I want to get across to you are, first of all, why antigen-specific immunotherapy, then the mechanism of antigen-specific immunotherapy with T-cell epitopes, and finally, I'd like to give you some examples of how we're taking this forward with three conditions: MS, Graves' disease, and the anti-drug antibody response to the clotting factor, factor VIII. So first of all, why antigen-specific immunotherapy? Well, and as I think it's become clear this morning, conventional treatments for particularly autoimmune diseases based on the use of antibodies to cells and cytokines tend to have side effects, many of which are really not acceptable in the long term. And of course, there is then the anti-drug antibody complication as well. So what we're doing here is targeting therapy to pathogenic cells in order to reinstate tolerance. Now, in effect, what we're doing is not at all new. And this slide dates back now 115 years old. And if only we could write papers with such wonderful language as this, the world would be a much better place. And this was before the term allergy was adopted in the UK. And when some of the older clinicians here may remember, allergy was known as a constitutional disturbance. But the truth is, if we were able to describe a clinical trial of a disease such as multiple sclerosis with these kind of results, then we would be very, very happy, okay? Now, the first question has to be the nature of the antigen, because of course, the reason that the approach that Freeman and Noon developed has been so difficult to adopt is because, of course, in allergy, intact antigen can cross-link IgE and lead to anaphylaxis. And when people have tried to desensitize patients with autoimmune disease, this has also led to complications through antibody induction and even cytotoxic T-cell induction. So many years ago, we started working with CD4 T-cell epitopes, and people like Mark Larche in London started doing the same in allergy. Then we spun apart and spun off companies working in allergy, in the case of Mark, and in autoimmune disease, in my own case. And I'm putting up this slide so that you should all go away and look up the papers describing the cat allergen trials from Circassia and the very, very remarkable long-term efficacy that they're finding. And if I remember rightly, the guys at the back will correct me if I'm wrong, but this is a 2-year follow-up. Yeah, they're nodding. So this is showing that a single dose or a single round of treatment with peptide epitopes appears to be sufficient to promote long-term tolerance. So can we apply this to autoimmune disease? Now, this morning, some of you will have noticed me referring to cryptic T-cell epitopes. And some people even ask me, what is a cryptic epitope? Well, I put this slide in just to illustrate this point. The truth is that some T-cell epitopes can induce tolerance, others can't, okay? And Steve Anderton, who was in my lab in Bristol a few years ago, decided to work on the one that didn't work, to try and figure out why it didn't work. Now, most people, of course, would work on the epitopes that do work, but Steve wanted to know why the one that didn't work. And he discovered it was because, having eluted the peptide out of the MHC, when you added it back to the MHC, it bound in a different conformation. It produced a different shape. It was a cryptic epitope. The peptide that bound back didn't engage the T cells reacting to the processed antigen. That's what a cryptic epitope is. So you heard a lot this morning about identifying T cell epitopes and about eluting peptides from MHC, etc., etc. This is a cautionary tale. This tells you be careful, because peptides don't necessarily bind back to the MHC in the same way as they would after processing of the whole protein. So you need to design peptides to mimic naturally processed epitopes, and we define those as antigen processing independent epitopes or apitopes. So that's where the name comes from. So what I want to do now is discuss work from my research lab at the university, where we've been studying how these things work. The APC they target, the mechanism of suppression, the mechanism of T-cell desensitization, and a little bit on IL-10. Now, this summarizes work from a PhD student in the lab, Ella Shepherd, and Ella has been looking at the antigen-presenting cells that soluble peptides target. So you inject a soluble peptide and you then isolate different subsets of antigen-presenting cells, and these peptides only target steady-state dendritic cells. You won't find them binding to B cells or macrophages. They're binding the different subsets of CD11C positive dendritic cells. And of course, in the steady state, those cells don't promote effective CD4 T cell differentiation, rather they induce tolerance. Now, why do those peptides target those cells? I believe it's because, as Laura Sant'Ambrogio showed, those immature dendritic cells, or the steady-state dendritic cells in lymphoid organs, have empty, or what I prefer to call peptide receptive MHC at the cell surface. So these are the cells that are being targeted. And then what happens? Well, Leona Gabrisheva was in the lab, and really she described a very important sequential effect of soluble peptide administration. That is, that in her model, Th1 cells, but this is equally true for Th2 and Th17, in her model, Th1 cells progress to producing predominantly IL-10. This cytokine then feeds back on local dendritic cells presenting the antigen, downregulate co-stimulatory molecules, downregulate MHC, and most importantly, block IL-12 production. And the block of IL-12 production then prevents further T helper cell or Th1 cell differentiation. So this is a negative feedback mechanism. And you see this mechanism actually in chronic infections. Now, we described this a number of years ago, and it's very gratifying to see a similar mechanism being described. This comes from Per Santamaria's group in Canada and Spain, and what they're showing is that nanoparticles coated with peptide MHC complexes directly drive this TL1-like phenomenon from Th1 cells. Now, they also showed that these cells mediate bystander suppression and block CD4 and CD8 T cell differentiation in a manner which is dependent on the different antigens that these T cells see being presented by the same antigen-presenting cell. So you can get linked and bystander suppression. And the other important thing about this paper was that it showed you didn't actually have to identify the dominant T cell epitopes for this phenomenon to be effective. Even subdominant T cell epitopes would work. So we're administering peptides that are binding to MTMHC on dendritic cells. They're bypassing that by making nanoparticles coated with MHC and antigen. But getting into the details of the mechanism of T cell desensitization took us into our T cell receptor transgenic model. This mouse expresses a T cell receptor specific for a brain protein, myelin basic protein. And if you give a dose escalation, you can get to very high doses of peptide which completely block spontaneous disease in these animals. You also induce anergy and IL-10 production, okay? Now, if you isolate the T cells from animals treated in this way and study them in vitro, you find that they form an immunological synapse, as shown here for CD3 and CD28. But most importantly, their downstream signaling machinery is uncoupled. So if you look at molecules like ZAP70, LAT, and PKC theta, in the tolerized cells, these molecules fail to form an immunological synapse, as a result of which the signaling through these molecules is blunted, and this explains both the anergy and comes partway to explaining the shift in cytokine production in these cells. But this is the desensitization that I was referring to. So, taking this model further, what we wanted to understand was what's going on at the cell surface to explain this T cell desensitization. And this is a summary of a microarray experiment in which we've looked at each sequential dose of peptide. And you remember this is during a dose escalation protocol where the animals are receiving every 3 to 4 days subcutaneous injection of soluble peptide, and then you're looking at the RNA expression in CD4 T cells. And there were two very, very striking sets of data from this experiment. The first was looking at transcription factors, and the biggest hits were molecules like cMAF, NFIL3, the aryl hydrocarbon receptor. And for those of you cytokine buffs in the audience, you will know that those molecules control IL-10 transcription. Okay? So some of the biggest hits in terms of transcription factors are factors controlling IL-10 production. You look at the secreted molecules and you see IL-10 and IL-21 RNA expressed precisely in coincidence with the increasing doses. But to us, a very interesting further observation comes from looking at cell surface receptors. And here you have molecules like LAG3, TIGIT, HAVCR2, which is TIM3, CTLA4, PD1, and of course, we all know these molecules now as negative co-stimulatory molecules or inhibitory receptors, because these are the targets that are eliciting such incredible excitement in the cancer field. These are the molecules that tumor-infiltrating lymphocytes are upregulating or being forced to upregulate, and this is why these cells are failing to eradicate the tumors. Now, I think it's interesting that if you look through the fundamental data of the trials of these molecules, of the checkpoint inhibitor receptors targeting these molecules, the very common side effect of these molecules is autoimmune phenomena, okay? Thyroid complications, vitiligo in trials of melanoma. And here I am saying to you, if you look at autoimmune disease, these are the molecules that are controlling the autoimmune phenomenon. Now this is the RNA, the real-time PCR validation of RNA expression. And this experiment here is interesting because this is doing the same experiment but in a IL-10 reporter mouse, and then looking at the expression of the inhibitory receptors. And in fact, the closest association with IL-10 producing cells, the IL-10 high cells, is not PD-1 or LAG3. These two are coming up on all anergic cells, but the closest association with IL-10 is TIGIT and TIM3. So I think these molecules, these inhibitory receptors, are extremely interesting, vitally important, but I think we need to be smarter to target these molecules in cancer while avoiding autoimmune disease. The other molecules that I referred to were the transcription factors, and this is a cartoon from a review by Sky in my lab. And Sky has now gone on to study the epigenetic changes in these IL-10 producing TR1-like cells. And NFIL3 appears to be having an effect by binding to exon 4, but CMAF is binding to the promoter region of the gene. And in CHIP experiments, Sky has shown that the peptide tolerance correlates precisely with downregulation of the repressive K27 mark in this region of the promoter. So it looks as though generation of CMAF is one required component, but opening up of the promoter region of the gene is also essential. In fact, we find that in naive T cells, the IL-10 locus is poised. Some of the repressive marks are there and some of the activating marks are there. And what happens when you differentiate a Th1 cell is that the IL-10 promoter locus shuts down, and then when we convert these into the IL-10 producing cells, the locus opens up again, okay? Coincident with upregulation of CMAF in these otherwise Th1 cells. So we believe that the experimental model that I'm talking to you about here has told us a lot about the mechanism of antigen-specific immunotherapy. We have a lot yet to learn, but some of the markers that we've identified promoting IL-10 secretion clearly become novel targets to promote tolerance induction. And the co-inhibitory receptors that we've seen clearly now become surrogate markers for monitoring effective tolerance in this system. So, now on to the work we're doing with Apitope, looking at anti-drug antibodies, and there I'm going to talk about inhibitor formation in haemophilia A. Then I'll briefly talk about Graves' disease, and finally talk about our work in MS, where we've done most of our clinical work. Now, possibly some of you in the audience won't be aware that the biggest problem facing people with haemophilia A is controlling their clotting if, sadly, they develop antibodies against the clotting factors. And we were just discussing at the coffee break, the truth is that about 20% of patients make antibodies to the plasma-derived factors, but unfortunately about 40% of patients respond and make antibodies against recombinant, the more commonly used clotting factors. So this is a serious clinical problem. Current treatments involve injecting tear-jerkingly large amounts of protein at vast expense. And it is true to say that most patients cannot receive this because of cost. The alternative strategy is to give high doses of bypass agents. We've picked out a couple of immunodominant peptides that according to the ProImmune Reveal assay are pan-DR binding epitopes. And this one here particularly is found eluted out of a wide variety of MHC proteins. However, these peptides do bind back to the MHC in the correct conformation and are therefore tolerogenic. And this shows a simple experiment where you give a dose escalation of peptide treatment, and this is in an HLA-DR positive factor VIII deficient mouse, and you can see that you pretty much knock out the T-cell response to whole factor VIII by this approach. But of course, this isn't how things work in real life. What we're challenged with here is a response to a clotting factor, which is a very large protein, but when given in weekly doses, is inducing antibodies, often in small children, with large deletions in their factor VIII gene. And in the factor VIII deficient mouse, you begin to see clinically relevant levels of inhibitor antibodies after about the fourth dose of protein. And you're getting very high inhibitor antibody levels after about 8 doses. So we follow this and look both for neutralizing antibodies and for inhibitor antibodies as measured in a clotting assay. And while there's a relatively modest effect on the total antibody response, as you can see here, the inhibitor response is being very strongly suppressed. And studies by our colleagues in Baxter, as it was, or Baxalta as it is now, or Baxalta Shire as it is now, have shown that indeed the inhibitory antibodies are the high affinity component. In other words, the T-cell dependent component of the total antibody response. So in a way, this makes sense, that by diminishing the T-cell help, you're losing the high affinity antibodies. So Graves' disease, this speaks for itself, and I think the real clinical need is to find a treatment that will deal with Graves' ophthalmopathy. We've designed peptides from the TSH receptor, and in HLA-DR3 transgenic mice, these inhibit the T-cell response, and using Basil Rappaport's adenovirus-induced antibody generation model, these also prevent the response to a significant degree. So finally, to finish up, and leaving plenty of time, hopefully, for discussion, I'd just like to cover the work that we've done in MS. And as you all know, the myelin sheath runs around the nerve axon, and in the myelin sheath, there are 3 dominant antigens: myelin basic protein, myelin oligodendrocyte glycoprotein, and proteolipid protein. We've designed epitopes from all of these proteins now, but the one we started off first with was myelin basic protein, and of about 8 clear T-cell epitopes within this protein, we identified 4 that were most commonly recognized in a large selection of MS patients. And these are all epitopes derived from T-cell lines specific for the naturally processed antigen. So we made a cocktail of these peptides and first studied them in the transgenic mouse made by Lars Fugger, where you have the HLA-DRB11501 DR2 molecule expressed, but also T-cell receptors from patients with MS, okay? And we induce disease, wait until the first signs of disease onset, and start treating at this point, and we're giving increasing doses of antigen here. And the 10 microgram dose in an allometric conversion is reasonably close to the dose we're giving to human beings. Clearly, this means that we could probably go to a higher dose, but what you find is a dose-dependent inhibition of disease. And these are peptides that are being given twice a week, so day 1 here, day 3, day 7, etc., etc. So this isn't continuous exposure to peptide, but repeated administration. So on the strength of this type of data, we do clinical trials, and this is the second clinical trial in relapsing remitting patients, 43 patients divided into 2 groups, intradermal and subcutaneous dosing, and we're giving the peptide every 2 weeks here, and the obvious primary objective is safety. The secondary objective, as part of the safety measurement, is gadolinium-enhanced MRI scanning and looking at the immunological parameters. But I want to focus really on the MRI scanning. So for those of you who don't know a lot about MS, the relapsing MS is a very dynamic disease. And this is looking at an individual over a 1-year period with monthly MRI scans. And you can see this inflammatory lesions cropping up, if you like, across the brain and then disappearing. So the gadolinium-enhanced MRI is measuring local inflammation. So we wanted to know what effect our treatment would have on new and persisting enhancing lesions. So we start a month before the first dose, measuring the MRI, and then at the first dose we start escalating up the dose, and it's at week 8 that you reach the high dose. So these patients are getting 800 micrograms of peptides subcutaneously once, twice, 3, 4, 5 times, and then you stop the treatment. And with time, the inflammation is coming back quite clearly. So this implies that to get effective cover, we would need to continue to give the peptide antigens. But we feel that this actually would be acceptable because so far, in about 70 patients that we've treated to date, we haven't seen any unexpected safety signals. There have been occasional skin reactions in the intradermal group, but nothing unexpected. So this implies to me that administration of these carefully designed innocuous peptide antigens, contrary to what many people thought, is actually a relatively safe procedure in humans. So what I've told you is that proteins can be replaced with peptides. This appears to be effective in a range of models of immune pathology. We've gained insight into the mechanism of action through the use of experimental animal models. And we are completing now a phase 2 in relapsing MS, and we're beginning trials with the hemophilia peptides and the Graves peptides this year. So finally, the people in my lab, particularly Bronwyn and Graham, who did much of the work that I was referring to, and the folks at Apitope who conducted the MS trial, and the people who've been developing the peptides for factor VIII and Graves' disease. Thank you very much.